Thank you. Good morning. A lovely, sobering kind of perspective there from Tamara, so thank you for that. It's our kind of privilege now to lead the first of the basic science sessions. My name's Ed Jarman. I'm a basic researcher up in Edinburgh, and I'll be co-chairing with Valerie Crowley.
Yeah, I'm a clinician at Barts in UCL.
Yeah, and the first of the basic research sessions is on specifically AMF-funded research. So the hope is that, or the celebration here is that we can look at all of the fantastic work that AMF is funding directly, and hopefully provide some level of optimism that things are moving forward and valuable research is making progress. So really looking forward to the basic science sessions.
So I'll start by introducing Ed, who is going to give his talk about does retinol from hepatic stellate cells fuel tumour cells during cholangiocarcinoma initiation? So take it away, Ed.
Yeah, great, thank you. I was worried about how I'd introduce myself in the session, so that was very facilitative, thank you. So as I say, it's a privilege to be able to come here and talk about some of the work that AMF is funding directly. This is a project that I've been working on collaboratively with another postdoc in our lab up in Edinburgh, Paula, and also it's generally a more collaborative effort than that. There's a number of researchers who are working together to try and understand this process. I'm going to talk today about retinoic acid signaling and its role in tumour initiation. And it's a work in progress, but we think it's a really, really interesting question, so hopefully I can lay that out for you and convince you of the same. So fundamentally, this really comes back to this question, which is something we're thinking about on a broader level in the lab. How does inflammation of the liver promote tumour initiation? So I guess it comes back to what's been discussed previously, that prevention is better than cure. And what we're trying to do is understand really early fundamental processes in tumour initiation so that we can identify, I guess, in the long term, potential ways of preventing this or identifying populations with inflammatory diseases that might represent a benefit from intervention of some form. So diseases such as PSC we're particularly interested in, but there's a number of inflammatory liver diseases that drive cholangiocarcinoma risk. And actually, the fundamental understanding of how epithelial cells with mutations harboring genetic aberrations are responding to these sort of inflammatory stimuli is still really not very well understood at all. So if I had to break our research down to some simplistic questions and summarize it, I would say this is what we want to ask. Do mutant epithelial cells misread inflammatory cues in the liver? And is this a fundamental part of how tumours initiate? Secondarily, and far longer term, our view on this is that if we can identify these processes, understand them, we might also be able to identify processes that we can get between and prevent, and find high-risk populations that we can treat in a way to prevent them from getting cholangiocarcinoma or reduce their risk. And there's also a potential for understanding mechanisms leading to more or better biomarkers for disease or disease risk. So our approach to this up in Edinburgh is very much using mouse models of tumour initiation, and we're focusing on a specific model that we quite like because it represents an inflammation-dependent tumour initiation model. So I'm going to just describe this briefly to you. We have a control mouse and a genetically perturbed mouse, and we, under the control of the keratin 19 promoter, we're able to drive recombination specifically in biliary epithelial cells. What that means in our control mouse is that we can drive the expression of this TD tomato fluorescent protein, and we can lineage trace the epithelial cells. That means from the moment of induction, we can follow these cells and isolate them at a later stage, and really focus down on these biliary epithelial cells that are becoming cancers in the mutant mouse. So in the mutant mouse, it's just a lineage tracing model. These ducts act very, very normally, but we can detect them through red fluorescent protein. In our mutant mouse, we also have floxed alleles of P53 and P10, and what this means is that when we induce this lineage tracing approach, we also lose expression of these two key tumour suppressor genes. So in this mutant model, they're poised for tumour initiation, but actually when we recombine in either of these and let the mice persist, histologically you don't see anything, we don't get tumour initiation, and actually very little of interest happens. What we need to do in addition is provide them with an inflammatory stimulus, and we do this through a compound called thioacetamide. So we give them thioacetamide in their drinking water. This drives inflammation. In our control mouse, we see a normal level of ductular regeneration. Here in the middle, in red, we've got the lineage traced RFP. These are the epithelial cells with the macrophages in green. This is just a standard H&E representation, and on the right is just a slightly more fancy 3D representation of these ducts, but essentially the ductular regeneration looks fairly normal. When we induce inflammation in the model that also has P10 and P53 loss, after about 3 weeks we get these early neoplasms forming. More clear here maybe in 3D. So to summarize, this is really useful for us. We really like this because it allows us to have an inflammation-dependent tumor initiation model, and it allows us to isolate cells from this very, very early on in the cancer kind of trajectory. So what we've done with this model is we've run single-cell sequencing. We've isolated the epithelial cells specifically, so we're very much focused down on epithelial cells in the hope that we can map heterogeneity across these cells and really understand the differences. So we've done this with 4 groups. That's our wild-type animals, those with inflammation, those with just mutation that don't get cancer, and then the combination of inflammation and mutation, which are a cancer model. And so we've been able to do some really nice comparisons and map the differences between these conditions, which I'm not going to get into too much depth today because that's the subject of an entire different talk and would take far, far longer than the time I have. What I do want to do is try and summarize a methodology that we've had that's allowed us to identify really, really interesting cell states through this data set. So it's a complex bioinformatics pipeline that's been developed by a brilliant physicist come biologist up in Edinburgh called Ava Kamsa. This is the publication that explains the pipeline that we've used, so I'd encourage anyone working with single-cell sequencing data to talk to me or have a look at this paper and assess this approach, because it's worked brilliantly for us in identifying cell states that were otherwise hidden, I guess, within our data set or hard to identify. So what we've done is we've used combinatorial gene modules to find rarer cell states. Rather than basing it on all of the variable genes or the 2,000 top variable genes within a data set, we're able to identify specific sets of genes that represent functional modules. I'm not going to, as I say, go into the detail, but what we can do is hierarchically cluster all the states that we get from this approach and end up in a place where we can say at what point in that clustering tree are these states specifically enriched in our tumor group and not in our mutant group. What this represents in a really simplistic way is that we have cell states that we would think represent pre-neoplastic mutant cells and the earliest neoplastic transition. So we can now make really, really refined comparisons in terms of gene expression differences at what we think is the initial trajectory of cells becoming very, very early neoplasms in this model. So this has been a really, really good tool, and as I say, I encourage anyone who's interested in actually understanding this and getting a fuller explanation of how it works, come and speak to me, because we're really, really keen for people to use this and to kind of spread the word that this approach is called Stator, is a very, very good pipeline for finding rarer states within these sort of data sets. But yeah, the main summary here is that we think we've identified pre-neoplastic and early neoplastic states that we can make very, very niche comparisons on. So yeah, this is something, really when it comes to, I've tried to keep this simple, and it's a slightly kind of broad interpretation of the data, but humanly. What we have really is large duct epithelial cells, or cells with more of a large duct phenotype, and we see a specific propensity for these to change their transcriptional behavior based on mutation. And these represent what we think is a set of cells that are perturbed by mutation and sit transcriptionally altered prior to tumor initiation. And then we have a set of cells that are only present when we have inflammation. So this is our early neoplastic set. So I'm just showing you where these sit spatially on the UMAP because that'll make more sense in the next slide. So the main question, why retinoic acid signaling? That's what I said I would talk about. From this data set and this investigation of the very, very early processes in tumor initiation, we, through gene ontology initially, retinoic acid signaling kind of rose up as something that might be of interest, and we looked into it in a bit more detail, and we found an example of a number of genes involved in retinoic acid detection and metabolism that seem to be specifically expressed within these same regions on our data set, so within our pre-neoplastic and early neoplastic sets. These include STRA6, RBP1, and RDH10. And so now I have the pleasure of very briefly trying to get you up to speed with retinoic acid signaling and how that works. So I'll give you a very quick overview of this, not to bore you. Essentially, retinol being transported extracellularly is an important signaling compound, vitamin A. It gets transported by retinoid binding proteins extracellularly, and STRA6 acts as a receptor for these proteins. So STRA6, this gene, is fundamental in cells detecting retinoids and bringing them into the cell. Once in the cell, it gets transported by intracellular retinoid binding proteins and goes through this pathway of metabolism where it goes from retinol to retinaldehyde to all trans or cis-retinoic acid. And this is done by retinol and retinal dehydrogenases, such as RDH10. Once we have our active ligand, which is either all trans retinoic acid or cis-retinoic acid, this binds to retinoic acid receptors and drives transcriptions of genes which modifies cell behavior in a very diverse number of ways that are quite hard to map, but we're thinking about this now. Importantly, this happens at promoter sites, which include retinoic acid response elements. But the important point I wanted to make about this slide is that the gene expression changes that we're seeing in our pre-neoplastic states are not necessarily transcriptional outcomes of active retinoic acid signaling. They're parts of the detection and metabolism machinery for responding to retinoids. So our kind of interpretation of this is that actually in pre-neoplastic states, cells are poised and ready to respond to retinoids in a way that's different to wild-type cells or non-mutant cells, which we think is quite interesting. So this is just zooming in a little bit more on specifically the cells that we think are pre-neoplastic poised mutant cells. In the greys here are our wild-type cells and our inflammation only, and this is a measure of expression of these genes. And as you can see, this goes up very, very strongly when mutations are introduced in blue and stays high when we introduce inflammation, so this is our tumour state. The important part of this is these are very, very high already prior to inflammation. So this is not a response to tumour initiation, this is not a response to inflammation, this is directly triggered by these mutations, P10 and P53 loss, and we think represents this interesting poised state. Excuse me. So the other interesting part of this is that we know within the liver, in response to inflammation, or prior to inflammation, I'll start, hepatic stellate cells are a cell type within the liver that store and hold on to retinoids. So there's a potential source of the initial ligands or the initial compound that gets metabolized into the ligands within the liver in hepatic stellate cells. Here I've got some immunofluorescence of fibroblasts with GFP in green, our epithelial cells in purple, and activation markers for these fibroblasts in yellow. So the green and yellow represents activated fibroblasts. What we know is that when hepatic stellate cells are activated by inflammation, they release these retinoid stores. Conceptually release them into the local environment, and they're there ready to be responded to by local cells. And we see that, obviously, this is probably common knowledge, but these kind of inflammatory fibroblasts are very, very closely interacting with tumour cells in this early tumour environment. Down the bottom here, I've just got in vitro indication that we can take out hepatic stellate cells, see via oil red, this is a lipid stain, that they store high levels of these lipids containing retinoids, and that when we activate them here artificially with TGF-beta, they release these retinoids. And on the left, these are activation markers showing that this is activation of these hepatic stellate cells. So what we think this represents is a combination of we have epithelial cells ready to respond to retinoids in a way that's driven by mutation and is very fascinating, and we have an environment where responsiveness to inflammation provides a source of this ligand. So we're wondering if this can in some way answer the question of why does inflammation drive tumour initiation. So our overall hypothesis on this is in that vein. Does retinoid release during HSC activation provide a source of retinoic acid for mutant epithelial cells? Excuse me. Excuse me again. Sorry. Mutant epithelial cells we think might be poised for retinoid uptake. This is really the interesting finding that we think we've landed on. And we really want to test, understand this, and see if this translates to retinoic acid responsiveness. And therefore, we think that retinoid availability during liver inflammation could be a driver of tumour initiation or could contribute to the process in some way. So how are we approaching this? At the moment, we're using organoid models isolated from these mice, and we've got our wild-type organoids, and we've introduced the same mutations. We've done this separately. We didn't want to perturb the system by isolating established tumour cells. We wanted this to be just a measure of how do mutations affect cell biology, so we introduced these via CRISPR. And actually, what we see, this is preliminary data to some extent, but we do see that upregulation of some of our markers for retinoid detection and metabolism are increased just through loss of these genes in organoids. So this supports the hypothesis that mutations are sufficient to drive changes in retinoid reception. So our approach now is we're developing a retinoic acid reporter system where we can use luciferase to understand as a direct readout for the intensity of the transcriptional results of retinoic acid signaling, and we're going to put them into our mutant and wild-type organoids to understand whether there's an actual functional difference in the downstream effects of retinoid signaling in these models. We've tested this reporter system in 2D cell lines, and we've shown that it's very retinoic acid responsive, which is good, so the reporter system's working, but unfortunately we're still in the process of creating these transduced organoids, which can be slightly finicky. We think we're there. We think in the very near future we'll have a perfect assay for answering the question of whether these mutant cells are responding to retinoids more strongly. So the set of experiments we're going to do in the near future is isolate hepatic stellate cells, condition media from their activation, and show that actually the retinoids secreted specifically from this cell type are driving, or ask whether they are driving, an increased retinoic acid response transcriptionally in our mutant cells versus our wild-type cells, and this will fundamentally help us understand whether mutations affect this response. The more fun, in my opinion, interesting experiment that we're going to do downstream, which I think is massively important, is to ask about physiological levels of retinoids and move back in vivo. And we've got this system where we'll be able to orthotopically inject these organoids, wild-type and mutant, into the liver of these mice, and measure luciferase activity in response to inflammation. So this will be a very, very nice model for really assessing whether the inflammation that is sufficient to drive tumour initiation leads to enough retinoids physiologically within the liver to change the responsiveness or to be detected in a more extreme level by mutant cells versus wild-type cells. And this will fundamentally support our hypothesis that this is a differential response in mutant cells that is driving tumour initiation. And we're going to isolate the epithelial cells from these orthotopic models so that we can really get a transcriptional readout for the pathways that are being changed and understand the specific transcriptional effects of retinoic acid signaling in this context. So that's kind of working from the start forward, trying to understand how retinoic acid signaling is working at a molecular level, how are these cells responding to retinoids, and are they driving an increased transcriptional response? At the same time, we're kind of working backwards and asking, can we just modulate retinoic acid signaling in our mouse model of tumour initiation and either reduce the level of tumour or stop it completely by antagonizing this pathway? So we've been concomitantly running a couple of mouse experiments where we've treated, alongside our KPP-TOM, this is our P10p53 mutation model, with inflammation from thiacetamide. So these mice get cancer at about 3 weeks, early stage tumours. And we've been treating these mice throughout that inflammation with an antagonist of retinoic acid signaling. This is canonical retinoic acid receptor inhibitor, AGN-193109, to ask whether this inflammation-induced tumorigenesis is dependent on this pathway. So really simple way of approaching the problem that we hoped might provide some answers of what's going on. Concomitantly, we've been doing the inverse, which is to upregulate retinoic acid signaling in these tumour models, but omit the inflammation. So these mice shouldn't get tumours because there's no inflammation to drive it, but if retinoic acid signaling is the fundamental pathway doing that, we can agonize it and maybe we would get tumours forming. So we've tried these two things. What we've learned is it's not that simple, unfortunately, which is science, and that's how science very frequently works. When we treat these mice with the antagonist for retinoic acid signaling, we see very, very similar levels of tumour burden. So here in brown, we've just stained once again for our lineage tracing fluorophore. So this is just a marker of the epithelial cells that have been recombined. And this is just low magnification and higher magnification of the same thing. When we measure the tumour area within these models, really we're not seeing any difference between agonist and control. So on the face of it, this is slightly disappointing, but really what it does is it educates us that this is not that simple, and potentially it's not canonical retinoic acid receptor response that's important. This inhibitor is a pan-inhibitor of the 3 main retinoic acid receptors, and inhibiting them doesn't seem to have an effect. So we're now drawing our sights directly towards some of the non-canonical retinoid responsive systems. And unfortunately, once again, I'm sorry to end my talk on a negative data thing, but I think it's very important to show this. And I'm being very genuine when I say these negative results have educated us quite quickly on what we think is going on. And so I think this is very, very good progress. But long story short, when we agonise with retinoic acid, with TTMPB, we don't see any indication that we're getting tumour initiation after 4 weeks or so. So it's not as simple as retinoic acid is signalling in these epithelial cells is driving tumour initiation. Upregulating that pathway to the max doesn't itself drive tumour initiation. The complexity of this pathway, which I didn't really want to go into today, is fairly extreme, and these processes are always very dependent on ligand concentration and physiological context and cell context. So our main thought on this is that we need to hone our sights on the other pathways that are being driven by retinoids, because our evidence suggests that these mutant cells are responsive to retinoids, but also shows the importance of testing this in a physiological level of retinoid availability, which is what we'll achieve with our luciferase organoid reporter system. So yeah, just to summarize slightly quickly, P10 and P53 loss in our epithelial cells seems to promote a poised responsive state. This is really what led to our interest in this pathway. And you know, as I say, this doesn't necessarily represent a transcriptional response to retinoic acid, but does seem to represent the genetic components that suggest they may respond better or more strongly to retinoids. Inflammation level could provide a source of retinoids through activated hepatic stellate cells, and we think this is really interesting because our fundamental question is all about how does inflammation interact with mutation, and this is one hypothesis for how that might be quite important. And so, yeah, we're working on this luciferase reporter assay, which seems to be working, but we're just in the middle of transducing it into our organoids. And this will allow us to do this really neat experiment where we can look at orthotopic injection and get a physiological level of retinoids to see if they're responding to that. And I can't stress enough how relevant retinoid and ligand levels are to activation of this pathway. And yeah, so the animal work so far, it's fairly preliminary. We're going to be repeating some of those experiments, and we're also going to be assessing whether these pathways were properly agonized or antagonized by our inhibitors and check out some pathways. But in practice, what it's hinting at is that this might not be a canonical retinoic acid response. This might be to do with retinoids driving other pathways, which have been shown in other contexts to promote cancer growth. So as a hypothesis, we're not too disappointed to be moved away from canonical signaling, but we need to work on this and understand this a bit better. Cool. And with that, I'll just kind of reiterate that this was definitely a collaborative effort within the lab. And thank you all for listening, and we'll do questions at the end.